Hello, and welcome to part two of our notes on cell organelles. Part two deals with cytoplasmic organelles in the endomembrane system. Let's begin. So, the first thing I want to talk about is the cytoplasm. Cytoplasm includes all the material between the plasma membrane and the nucleus, everything, the liquid stuff, the organelles, the bits of things floating around, which is a little different than what you learned in bio. In bio, we said it was the liquidy stuff between the cell membrane and the nucleus. We're getting a little more specific now here in Anatomy Phys. That liquidy stuff technically is called the cytosol, mostly water and various dissolved proteins, salts, sugars, and other things just dissolved in that fluid floating around in there. Then we have the cytoplasmic organelles are also considered part of the cytoplasm. So those would be things like the mitochondria, the ER, the Golgi apparatus, the ribosomes, those types of things. And the last piece we have in there are inclusions, chemical substances such as pigments and other bits and pieces floating around in there. All right, next we have those cytoplasmic organelles, where the main focus of this series is on. So these are specialized cellular components. I mean, this is just review from bio, right? They're membranous, so we have membranous versions of them, but we also have, which include the mitochondria, lysosomes, the endoplasmic reticulum, and Golgi apparatus. Hopefully a lot of those you remember from biology already. And then you have non-membranous ones, which some of these are going to be a little bit new that we didn't really talk about much in bio. Ribosomes for sure, cytoskeletons, and centrioles, which we'll get to later. Let's start with the mitochondria, the powerhouse of the cell. It is a double membrane structure with shelf-like cristae folds, which are folds, for more surface area. I'm going to skip ahead to the next slide just to show you that. So we're taking a look in here, guys, and we can see, oh, it's covering it up here. Down here, we can see here's a mitochondria, all these folds in the middle, and those folds are studded with enzymes. All these little balls here, those are enzymes, and those enzymes are what carry out the functions of the mitochondria. And by putting them, basically you can think of them on a sheet like this tissue, right? Full of enzymes, which is great. This is where all the cellular respiration is going to take place. Now, this takes up a lot of space in the cell, right? So what you get by doing all those folds inside the mitochondria is we take that same amount of surface area, the same amount of chemical processing power, and we ball it up into a much smaller volume, but with the same surface area. So we can fit a lot more enzyme action into a smaller space, and it's much more efficient this way. So that's why we have that particular physical arrangement. You're going to see this all over our study of Anatomy Phys. Surface area is king. We see it here. We'll see it in the lungs. We'll see it in the digestive tract. We'll see it in capillaries. Surface area plays a huge role in how our body works. And when you see these crumpled up surfaces, the reason we're doing that is for surface area. And we'll get into that more as we get into those other systems. Oops, sorry, got to go back. Now, the reason we need that surface area is because this is where our cells get most of their ATP, adenosine triphosphate, which, if you recall from biology, is the fuel for our cell. Obviously, we take in things like glucose, and we can break those chemical bonds to release the energy stored in those chemical bonds. But that energy from glucose, our cells can't use directly. It's kind of like, you think of it like coal in a power plant, right? We can't use coal to power our toasters or our refrigerators, right? So the power plant is like the mitochondria. It's going to take that raw material, okay, that coal in the case of a power plant or glucose in the case of the mitochondria, and it's going to convert that energy into a useful form. And for our cells, that useful form is ATP, adenosine triphosphate. The energy stored in that is small enough, compact enough, and usable by all the different cellular machinery we need. So the mitochondria takes that glucose, turns it into ATP via a process called anaerobic cellular respiration, which is our main source of energy. We can do anaerobic without oxygen, but it's much less efficient. We'll talk about that when we get to the respiratory system. But anaerobic respiration is the main source. And interestingly enough, mitochondria contain their own DNA and RNA. And there is something called the endosymbiote theory on why that is. The mitochondria have separate DNA from your chromosomal DNA that's in your nucleus. Okay, they reproduce on their own. Okay, and you'll hear about this sometimes in the news. Somebody will talk about mDNA or mitochondrial DNA, and being able to trace back your maternal lineage via that, because all the DNA, all your mitochondria came from your mother's egg. Those mitochondria were in your mother's egg, and when that egg cell divided, so did those mitochondria, and they migrated to all your other cells throughout the process of cell division. And it's pretty cool. We think the reason that is the case is way, way back in evolutionary history. We had these, big cells that, and we have these little bacteria there, mitochondria, that were really good at producing energy. And make a long story short, some of those bacteria that became mitochondria wound up inside these bigger cells, and they found a symbiotic relationship, endo-symbiote, inside symbiote. Um, and this relationship, the bigger cells provided the materials and the protection to those smaller bacterial cells. Those bacterial cells were able to more efficiently produce ATP energy for the larger cell, to the point where they became interdependent, and we consider them all one cell now, one organism. Though the origin is probably two came together to form one, which is pretty sweet when you think about it. All right, ribosomes. These are little granules. They have protein and rRNA, or ribosomal RNA, makes them up. And the main thing you got to remember with ribosomes is they're the site of protein synthesis. This is where we make proteins. These are the units that take all these separate amino acids, the monomers of proteins. There's 20 different ones of these, and bind them together in a particular order, like the different colors on this chain, which will make a protein strand. And based on the properties of these different amino acids, that protein strand will fold in a certain three-dimensional pattern, right, to make a 3D object. And these proteins then become the molecular machinery of our bodies, of all life on Earth. They make things like antibodies that have the right shape to latch on to, say, coronavirus, if you get coronavirus antibodies produced, and take that out, or hemoglobin's got the right shape to grab onto oxygen and carry it around your body more efficiently, or enzymes. They have the right shape to catalyze various chemical reactions and make them happen more efficiently and faster, and where you want them in the case of aerobic cellular respiration in the mitochondria. So these proteins are extremely important. This is what DNA codes for. DNA codes for making these proteins. These proteins then control everything else to make us us. And all this is made at the ribosome. Now, ribosomes you'll find them in two different places in the cell. One, you got free-floating ribosomes like we've got down here. Oh man, I hate that this thing keeps popping up. Can I move that? I cannot. But underneath where that thing is, there are some ribosomes just floating around free on their own. Okay, and those ribosomes will produce proteins that what we call soluble. They'll be able to go wherever. And then there's ribosomes that are membrane-bound ribosomes, which you see here attached to the endoplasmic reticulum, which is labeled number 2. And those membrane-bound ribosomes are going to make proteins generally that are going to be bound up in membranes, like we talked about in the last section of notes with the cell membranes. Got a lot of proteins embedded in there, do a lot of different jobs, or those enzymes embedded in the mitochondria. That's where these enzymes or proteins are going to get produced, is if they are on this membrane of the endoplasmic reticulum. Speaking of the endoplasmic reticulum, they're basically a series of interconnected tubes and parallel membranes. Yeah, like a big old subway network almost. They are contiguous to the nuclear membrane, meaning they in some places actually hook up with the nuclear membrane. And there are two main varieties, rough ER and smooth ER. And we'll talk about why they're called rough and smooth in just a second. Here on this picture, we can see here is a nuclear envelope, double membrane nuclear envelope, and then the blue stuff here are the ribosomes. Can I make that thing go? Yeah, right here. Or the blue stuff is the endoplasmic reticulum, sorry. Here and here. Now I notice they look different. Down here, this is rough ER. Rough ER is so called because you can see all those red dots. Those are the um, um, ribosomes embedded in them, making it look rough, like sandpaper almost. Um, and then over here we have smooth ER, more tubular in shape generally, and no ribosomes, so it's smooth in appearance. And they serve different functions. They have a different structure, and those different structures allow them to do different functions, which is another common theme we're going to see in Anat and Phys. Uh, Form follows function, and function determines form, oftentimes. So, the rough ER. Got a little picture of it down here. The external surface is studded with ribosomes, hence it's called rough ER. And it manufactures any proteins that are going to get secreted out of the cell are going to be made here. The ones that are made by the free-floating ribosomes are going to stay in the cell. But if you're going to make them and ship them, we're going to want to make them in a concentrated place where we have control over them, so we make them in the rough ER. It is mainly responsible for the synthesis or production of integral membrane proteins, meaning proteins that are bound up in a membrane, and the phospholipids, the membrane that those proteins are bound up in. So it basically makes like cell membrane or mitochondria sheets and things of that nature. Here we got smooth ER. Now these tubules are arranged kind of in a looping network we can see in that image, and they catalyze or speed up the following reactions in various organs of the body. So based on what tissue and organ they're in, they're going to do slightly different things for us. But essentially, they all have to do with lipids and lipid production. So whereas the rough ER dealt with membrane production with embedded proteins, smooth ER generally deals with lipids. No ribosomes on it, so it's not dealing with proteins, it's just dealing with lipids or fats. In the liver, it helps with lipid and cholesterol metabolism and breaking down glycogen, also some detoxification and things of that nature. In the testes and ovaries, it'll be involved in the synthesis of steroid-based hormones like testosterone and estrogen. In the intestinal cells, down in your gut, it's going to help with absorption, synthesis, and transport of fats in those areas. Once again, more lipids. Steroids, by the way, are a lipid-based hormone. And in skeletal and cardiac muscle, helping to release calcium and control the levels of calcium, which is huge for how those function. We need very precise amounts of calcium in muscles in order to get them to function properly. And we'll talk about that when we get to the muscular system. Next, the Golgi apparatus. In biology, we may have used the analogy the Golgi apparatus was like the UPS store, right? You bring in your packages and all your various things, and they'll sort them, they'll package them, they'll box them up, and they'll ship them out where they need to go. Basically, what the Golgi apparatus does. They're kind of stacked, membranous stacks, sacks. Sometimes people liken them to looking like a stack of pancakes. They function in the modification, concentration, and packaging of proteins and other cellular products. Okay, so modification, they'll take like a couple different ones and stick them together. They'll concentrate them, kind of build them up so you get a nice tight bundle to ship. You don't want to ship out 800 little packages. You'd rather ship out just one big package with everything on there. And then just packaging them in general. So the way this works, and I've got an image on the other side, but we'll go through the words here on the next slide. Transport vesicles from the ER, from the Endoplasmic Reticulum, that are holding all the stuff the ER made, fuse with the cis, which means near face of the Golgi apparatus. So the face of the Golgi that's closest to the ER. And the ER is usually closer to the nucleus, and the Golgi is a little bit closer to the surface of the cell, a little more superficial to the cell membrane, or to the ER. The proteins and other products will pass through the Golgi apparatus where it's modified or packaged or concentrated, whatever it is it needs to do in there, to the trans face or the far face of the Golgi, the face closest to the cell membrane. And then little vesicles will butt off from that trans face and move to different parts of the cell where they're needed, or to the cell membrane so we can release them out into the world. Here we can take a look at that. Here is kind of a Golgi. This right here is the cis face, so the ER would be somewhere over here, up where the Golgi apparatus words are. So the material is going to come in. You could think of this like a Oh, like a car factory. Car factories don't make every single little part for the car, right? They make they put all those little parts together to make the final product. So all these different parts are going to come into this side. This is the receiving end of the factory, right? So you get your engine parts, your body panels, your suspension, your seats, your wiring, all coming in this side, all separate, made somewhere else in the ER or whatever. They're coming in, and you can see these vesicles will fuse with the near face, the cis face, and release their products into the Golgi. And then as they work their way through the Golgi, that's like working its way through the factory, right? You're going to take all these various parts and you're going to put them together to make the final product. And then you're going to package all these final products together and ship them out. So here we've got one. This would be all those different cars packaged together on a car carrier or a ship or something to take wherever they need to go. We have some other ones though that will stay in the cell. Okay, so you can think of that like car factory. You got a lot of maintenance stuff that needs to happen, right? You got to build the machines to build the machines. You got to repair them. You got to fix them. So what you're also receiving over here is the parts for keeping the factory running, and the Golgi apparatus will put those together and send them elsewhere in the cell so the cell can keep working. And that's what the Golgi does. Here we can kind of see a little bigger picture. So we got the ER making its stuff, these little vesicles popping off, carrying the material over to the Golgi. Golgi's packaging, putting them together, modifying whatever it needs to do. Here's one that's collecting up all of the stuff that's going to get shipped out of the cell, kind of butting off this little, kind of like pinching off a little bit here in a little transport vesicle, transport out to the cell membrane, and you can see how it fuses with the cell membrane here and releases all this stuff out of the cell in one big batch. Over here we can see one that's actually going to be cell membrane. It's got all this stuff. It's going to be incorporated in the plasma membrane. And over here we have one that's making those internal parts for the cell and keeping that right there. Lysosomes is one of those internal membranous organelles that the Golgi makes. And basically the lysosome is just a sac containing digestive enzymes. We often refer to this as like the recycling center of the cell, right? It's going to digest ingested bacteria and virus and toxins. So if there's stuff you got to break down, this is the organelle that's going to do it. It's full of all sorts of digestive enzymes in there. When organelles break down or don't work anymore, you got to get rid of them. But you can recycle those bits and pieces. So the lysosome will break them down into their constituent molecules. Non-useful tissues. They are a big part of the cells, osteoclasts, and we'll learn that later, that break down the calcium portion of bones, the hard portion of bones, to release that calcium back in the bloodstream. They literally eat away your bones because your body thinks it's more important to have the right amount of calcium in your blood than it is to have strong bones on a minute-to-minute, day-to-day basis. You want strong bones, but if you don't have calcium, your nervous system and your muscular system will stop working. So these are important for that as well. And just putting it all together, most of those organelles we talked about, we call them the endomembrane system. It's a bunch of organelles that function to produce, store, and export biological materials. Get rid of harmful substances like we talked about with the lysosomes. And it includes all those organelles that we've just talked about so far in the first couple sections of these notes. So we can see here, the nucleus, it's going to contain the DNA that's got the instructions for making the proteins. So you'll get DNA, you'll make a copy of a gene onto mRNA, messenger RNA, which will send it out to the various ribosomes. And these various ribosomes will then make the proteins. If they're just free-floating ribosomes out here, there's going to be proteins that stay in the cell. If not, they're going to come to these ribosomes in the ER. The ER is going to make those proteins and plasma membranes and all that kind of stuff. Some of that's going to just stay in the cells. A lot of it's going to ship to the Golgi, which will package it up and turn it into other things and then ship it out of the cell. And that is it for part 2. Join me later for part 3 on the cytoskeleton and the nucleus. Thanks. Bye.